All right, at this point, I'm going to turn over to the audience questions. First up, we have a question. Could you address the size of GOI? Some organizations are using nanoplasmids to carry large payloads. Steve, if you could take that question first, perhaps help Abe.
I didn't see it written, but I think I know what you're saying. So in terms of payloads, and I think what they're referring to is, say, maybe a gene insertion. At Caribou, we're using adeno-associated virus, particularly isotype 6, because it infects T cells very well. It doesn't happen to work as well on iPSCs because they don't have the receptor. And so it really boils down to what cell are you targeting? Does it have a receptor for the virus that you want to use? Now, again, going back to lipid nanoparticles, they're pretty much limited to liver. They do go to other, or you'll find them in the kidney, and you'll find them in the lung. So when you blast them into the body, they will go to different places. And as we mentioned, you could try to ligand them in different ways to sort of bias them to go to different tissues. But I think, there are limits, right? People use lentiviruses for making CAR T cells, as for instance. It has maybe a little bigger capacity for a larger gene or maybe two genes. AAVs are pretty restricted to about 3.5 kb, in addition to the 1,000 nucleotides that you need to keep there for the virus to work in the first place. So every system has its limits. And I think, as we get to non-viral methods, depending upon the way that you could modify the oligo, which could be a plasmid, it could be single-stranded, it could be different things, we could begin to make much bigger payloads. And I think that is one way through. And I'll leave it to Eric to finish.
Yeah. Eric, feel free to comment.
Yeah, don't have too much to add to that. I think you can look at what's the size of the payload that you're looking to target, and then look at what the capacity for the viral vector or nanoparticle, whatever the carrier is that you're using, and just determine the correct size based on that. So yeah, all the items that Steve mentioned there and the size restrictions, I agree.
Good. Okay, thank you. Just a couple more questions that came in as we're talking here. What would you say is the best software to detect off-target effects? And I'll open up this to either one of you, whoever would like to take it first, Eric or Steve, feel free. Any software tools that are good for this purpose?
Yeah, I mentioned a number of them earlier, right? I think that can be considered, and they all have slightly different characteristics based on, the speed that, you want to transfect your cells in vivo versus ex vivo, but or cell-based strategies to detect off targets. I think, there's not one specific tool that I think is best, but I think there are some pretty good review papers and other aspects on this. I mentioned Deep CRISPR is interesting because it can take into consideration the epigenetic characteristics. Other ones that are other techniques that use cell-free versus cellular context for identifying those edits. So it does come down to your research question and what you're looking at. But I've been interested in some of the ones that, like Bless and Bliss, that can directly identify those breaks in the DNA versus targeting and enriching certain areas. I do think as whole genome sequencing prices come down more and more, it's like Steve mentioned, flashlight in the dark, the larger area that you can sequence and identify double-stranded breaks will be good to cover your bases and make sure you're not missing something because you're not looking. So I guess I'll go ahead and leave it there. I can hand it to Steve.
Yeah, I think that really covered a lot of the bases. If you're doing some deep sequencing, maybe not whole genome, but at least at the sites that you believe where off targets may occur, a lot of the systems for those, I unfortunately can't mention what we're using, but they come with their own software, and these are all commercially available systems.